Good evening, Sudeshna. Good evening, everyone. Good evening, Aditya. Good evening, Dolly. Good evening, Bhavya. Good evening, Aditya. Good evening, everyone. We will start the class at 7, okay? Bhavya, there is no need to give your attendance. Your attendance is automatically getting recorded, okay? Good evening, Divya. Let's wait for 2 minutes. Sakshi, Mukesh, Aditya, Mahadeva, your attendance is getting recorded, okay? So there's no need to write in the chat box. Your attendance is getting recorded. Okay, so let's start with our today's session. Yesterday, we worked with only one DNA sequence. But in real bioinformatics, we never analyze just one sequence. We work with hundreds, thousands, sometimes millions of sequences. So today, we will learn how to handle multiple sequences using Python. So imagine that you are given 100 DNA sequences from different samples. Now, if we store them in separate variables, that means we will have to make 100 different variables. Now, it becomes really messy and it is almost impractical. So, to make 100 variables to store 100 different DNA sequences, this approach is really bad. So what we should do instead? We should make a list. Now, what is a list? A list is a collection of multiple values stored under one name or under one variable. Just like a folder contains multiple files, similarly, a list can contain multiple values. For example, let's open our VS Code. Here, I am making a variable sequence, and inside this variable I am storing a DNA string. So here I am storing ATGC. So here, as we can see, that this is a single DNA sequence or this is a single string. So I am storing a single value inside my variable sequence. Now, what if I have two or three other DNA sequences? Now, just like we discussed above, that making multiple variables to store multiple DNA sequences, this idea is really bad. So, what we should do instead? We should make a list. Now, how to make a list? Here, let's imagine that I have one more DNA sequence. So, here I will just add comma, add quotation marks. We also call it double quotes. Now here I will write my DNA sequence. Suppose I have GGCTA. Now add comma to add another DNA sequence. Suppose now I have TTAAC. So what I am doing here, I have added three different DNA sequence. This is DNA sequence 1, this is DNA sequence 2, this is DNA sequence 3 separated by comma. But this is still not a list. Here it is still not a list. So how to make it a list? We write it inside square brackets. So let's write it inside square brackets. Now this is a list. If we write square brackets and we add our multiple values inside it, this means that we have stored multiple values or we have stored multiple DNA sequences inside a list. Now we can also make Python to print the type of our variable. We have already learned how to print the type of our variable. We write print, then add parenthesis. Now here we write type, then add parenthesis again. Now inside this parenthesis, write the name of your variable, which is sequence. Now let's run this. So here Python has printed us class list. So Python also knows that this is a list. So here each DNA sequence is still a string and the list simply groups all of these strings together. Now what if Okay, now just like we learned indexing yesterday, that to access a specific character in a string, we use the index number. Similarly, we can also use indexing in a list. Indexing rules are the same, that counting starts from 0 in Python and not from 1. The only difference is that in the string, here, this is a string. So in the string, we learned that each specific character has a specific position number or an index number. But in list, these are multiple items. These are multiple values. So in list, we do indexing like this, that this item is stored at index number 0, then this item is stored at index number 1, and then this item is stored at index number 2. So here our first DNA sequence is stored at index number 0, our second DNA sequence is stored at index number 1, and our third DNA sequence is stored at index number 2. Now, just like we used the index number yesterday to access the specific nucleotide in the string, in the same way, to access a particular item in a list or to access a particular DNA sequence in a list, we will use indexing. So, just like we learned yesterday, we write print, then we write the name of our variable, then we add square brackets. Now inside the square brackets, we give our index number. So let's write 0. Here I have given the index number 0. Now when I will click on the run button, Python will print me ATGC, means the item stored at index number 0. Let's run it. So here I am seeing ATGC because it is stored at index number 0 inside this list. Let's change the index number. Let's write 1. Now, by giving the index number 1, I am trying to print the second item of the list, which is GGCTA, my second DNA sequence. Let's run it again. So here we are saying that Python has printed us the value stored at index number 1. Similarly, if I write index number 2, Python will print me TTAAC. Let's run it again. So we are seeing TTAAC. Now, let's understand one very important concept that many of you can get confused about. We are going to talk about two different types of length. We are going to talk about two different types of length. One is the length of the list, the length of this whole list. And the second is the length of a DNA sequence. Now, these two lengths are not the same. For example, we know that this variable sequence is a list because the value which we have stored inside this variable is a list. So the type of this variable is a list. And this list, inside this list, we have stored 3 DNA sequences. So if I write print, then I use my length function, add parenthesis, and here I give the name of my variable sequence, and now let's run it. Now here we are seeing 3, which means that this line does not count how many nucleotides are present here. It only counts how many items are present inside this list. So here in the output we are seeing 3. So this means that this line print length of our variable sequence, it means that it has printed me the number of DNA sequences stored inside this list. Okay. Now what if I write print the length function, add parenthesis, write the name of your variable. Now add square brackets because inside the square brackets we give the index number. Now here I write 0. Now this 0 index number means the first DNA sequence inside this list, and by writing this whole line, what I'm trying to do here, it means that we are trying to print the length of our first DNA sequence. Let's run it. See, so this line has printed me 4, which means the value at index number 0, the value of my DNA sequence. So here the length of the DNA sequence is 4. If I change the index number to 1. Now let's run it again. So here in the output, we are seeing 5, which means the length of my this DNA sequence is 5. Let's count it 1, 2, 3, 4, 5. So we are seeing that by writing this line, we are trying to print the length of our DNA sequence. So this line tells us how many DNA sequences are in this list, and this line tells us the length of one DNA sequence. So in this topic, we learned that the length of a list, here the length of the list tells us how many sequences we have, and the length of the sequence tells us how many nucleotides are in this DNA sequence. So by 5, it also means that we have 5 nucleotides in this DNA sequence. Now till here, do you have any doubt? Anyone has any doubt till here? Okay, Vidasri, very good. Does anyone has any doubt? Do you want me to repeat anything? Okay, very good. Yes, Azmi. Square brackets again. Okay. See here, yesterday we learned about indexing. Let's write simple. Okay, let's write print and then we give the name of our variable. Now by writing this line here, we are giving the instruction to print the value inside our variable. So by writing the name of our variable, this is our value. Now when I will click on the run button, let's run it. So it has printed me the value which I stored inside my variable, and this is a list. Now here I am trying to access these items. I am saying that this is a list and these multiple values are called the items inside the list. And we know that this is a DNA sequence. Now what if I want to access this DNA sequence? So here we use indexing. So we learned yesterday that in Python, counting always starts from 0. So this DNA sequence, the first item of this list is stored on the index number 0. Then the second item of the list is stored on the index number 1, and the third item of this list is stored on the index number 2. Now, how to access this DNA sequence? We will add square brackets here, and inside the square brackets we will write our index number. If I want to access this DNA sequence, I will give the index number 1. So let's write 1 and now run it. So here we are saying that Python has printed us GGCTA because inside the square brackets we have given our index number 1 because we want to access this DNA sequence. Is it clear? Azmi, is it clear? You write the index number inside the square bracket. Okay. Now we learned that we can store all the DNA sequences together in a list. But here we have taken a very little example. We have only stored 3 DNA sequences in a list. But what if we have many sequences? So if I try to print them one by one using the print statement, it will become really messy. So I want Python to take each sequence from this list and print it. But I don't want to manually give the index number like sequence 1 or sequence 2 or sequence 3. So I want Python to do this automatically. So for this, we use something called loop in Python. So here I'm writing a code for S in sequence colon, press enter, write print, add parenthesis. Now inside this parenthesis write S. Now what does this code mean? What is this code actually doing here? Let's understand this very slowly. We already have multiple DNA sequences stored together in a list and inside the variable sequence. Now here our goal is very simple. We want Python to print each DNA sequence inside this list one by one automatically. So for this, I have written this line for S in sequence. Now, this line is telling Python to look inside the variable sequence. Here, we know that sequence is a variable. So here, by writing this line, we are telling Python to look inside the variable sequence, where we have stored a list. And here, S. Here, S is just a temporary variable that I have given to Python. So Python will use this temporary variable to store one DNA sequence at a time. Now when I will click on the run button, what will happen? Firstly, Python will take the first DNA sequence from this list and will store it inside the temporary variable S. And after that, I have written the line print S. So this means that Python firstly will take the first DNA sequence, it will store it inside the temporary variable S which I have given to Python, and then it will print me the value of S. So currently the value of S is ATGC. Now Python again will go back in this list. Now Python will take the second DNA sequence, it will store it inside the temporary variable S and it will print the value of S, which means in the second line of our output, we are going to see GGCTA. Now again, Python will go back inside this list. This time Python will pick up the last DNA sequence, it will store it inside the variable S and then it will print the value of S. Now this time the value of S is TTAAC. First of all, let's run it and check our output. Now here we are seeing that in the first line of our output, Python has printed us the value of S which was ATGC. Then again in the second line of output, we are seeing GGCTA, which was our second DNA sequence. And lastly, Python has printed us the last DNA sequence. Now this is called loop. This is called loop. Now, when there are no more sequence inside this list, the loop in the Python has stopped. Okay, so here Python is not printing the whole list together. It is printing one sequence at a time. So this is the loop that we have created by using the for keyword. Here for is a keyword. So whenever we want to create a loop in Python, we will use this for keyword first, then we will give a here, then we will give a temporary variable. We can name it anything. I can write it P, I can write it X, I can write it whatever I want. I can also name it animal. Whatever I want, we can give any temporary name we want. Then we will write in, and then we will give the name of a variable inside which our value has stored. After that, we will add colon. After adding colon, here, after adding colon, we will press enter and we will write print, and inside the parenthesis, we will give the name of our temporary variable, which is x. Now let's run it again. So this is called loop. This is the complete syntax to create a loop using a for keyword. So whenever we use for keyword to create a loop, it is called a for loop. So here I have created a for loop. So what is a for loop? Whenever we want Python to repeat the same task multiple times automatically, it is called a for loop. Is there any doubt regarding this loop topic? Okay, let's take another example. Okay, let's take another example. So here, just like we created a variable sequence, here I'm going to create another variable called genes. And now inside this variable, I am going to store a list. Now we already know how to make a list. We use square brackets. Now inside the square brackets, we can store our multiple values. So let's store a gene name BRCA1. Let's store another gene name TP53. Let's store one more gene name EGFR. So this line is clear that we have created a variable genes, and inside this variable we have created a list, and inside this list we already know that this is a string. Anything we write inside the quotation mark is a string, but we know that these are gene names. Now I want Python to print all the values inside this list automatically. Suppose if I want to access TP53, I do not want to do it manually. I do not want to access it using the index number. I want Python to print me everything inside this list automatically. So whenever we want Python to do these kind of things automatically, we create a loop. We already know what a loop means something is repeating. Now here we learned a for loop. Here we learned for loop. There are actually two types of loop. There is a for loop and there is a while loop. But today we are only learning about for loop. So here we learned about how to create a for loop. To create a for loop, we have a keyword called for. So whenever we want to create a for loop, firstly we will write for. This is it. To create a for loop, we will use the keyword for. Now after using the keyword for, we will write a temporary variable name. So here I am writing G. Simple, I'm using a simple temporary variable name. I have written G. Now, where I want to use the loop, where I want Python to use the loop, I want Python to use the loop inside this list. So what will I write here? Here I will give the name of my variable genes. because this list is stored inside my variable genes. So I will write in genes. Okay, so here I have given the name of my variable. Then we will add colon. This is syntax. You have to write colon at the end. Now press enter. So here we are saying that after pressing enter, there is some space, right? After we add colon at the end and then we press enter, there is some space. So this is called indentation. The student who asked about indentation earlier, this is called indentation. It only means that we are giving some space. Now here in this line, we have to write print because we want to print in the output, right? We want to see all these multiple values in the output. So we have to write print. Now, what we want to print, we have to write the name of our temporary variable which we created here. So let's write G. Let's comment it for a second. Now our program is complete. We just have to click on the run button. But before clicking on the run button, let's just understand what this program will do. And by this program, I mean to say what this loop will do. Now, when I will click on the run button, what will happen? Here I have written for means I have created a loop for G in genes. Means firstly Python will go inside this list and the first gene name is BRCA1. It means Python save this gene name inside this temporary variable G. Now the value of G is a variable, it is a temporary variable. Now the value of G is BRCA1. So now when I will click on the run button, in the first line of my output, I am going to see BRCA1. Now the loop will run again because we have two more values inside this list. Now the loop will run again, means Python again will go back inside this list. Next time Python will see that there is another item TP53. Now this time Python will save this gene name inside this variable G and it will print me the value of G. So in the next line of our output, we are going to see TP53. Now again Python will go back inside this list because there are one more item left. There is one more item left. Now this time Python will put this gene name inside this temporary variable and it will print me the value of G. So in my third line of the output, Python will print me EGFR. Now there is no more item left inside this list. It means the loop has ended. So in the output, I am only going to see all the items present inside this list. So here in the output, we are seeing all the items which were present inside our list. And we also understood that Python is not printing all of this all together. Python is printing it one by one. In the first loop, the value of G was BRCA1. So here we are seeing BRCA1. In the second loop, the value of G was TP53. So in the output, we are seeing TP53. Then in the third loop, the value of G was EGFR. So in the output, we are seeing EGFR. Now, as all the items has ended in the list, the loop ended. This is called a loop. Is it clear? Do you want me to repeat it again? It is very simple. It's not that much difficult. Yes, Vidasri, it is printing one item at a time. We can also take a very simple example. Here we are taking the biology related example. We can also take very simple examples. Like here, let's create a very simple variable named fruit. So I have created the variable named fruit. Let's create a list here. We have added the square brackets. Now I am adding some items. I have added apple. I have added banana. I have added mango. Now I want Python to print all the items of this list. But what did I just say? That I don't want to do it manually. I want Python to do it automatically. I want Python to print all the items in this list in my terminal. So we will create a for loop. So to create a for loop, we will use the for keyword. So firstly, we have to write for. Then now here, we will give a temporary variable name. So here I'm giving f. Then in fruits, because I want the loop to run in my fruit variable. So here I have written fruit, then add colon, press enter. Here this is indentation, it is called space. Then write print and give the name of your temporary variable f. Now the same thing that happened here, the same thing will happen here as well. There the loop will run. In the first loop we are going to see apple, in the second loop we are going to see banana, and in the third loop we are going to see mango. Let's run this as well. Let's run it. So here in the output we are seeing that Python first has printed us apple, then it moved to the second item, it saved this value inside the temporary variable f, and it has printed us the value of f, which is now here banana. Then the loop run again. Here Python saved this value mango inside the temporary variable f and has printed us the value of f. So here Python is not printing this list all at once. It is printing it one by one. Okay, so is it clear? So the loop will run only one time. See here in this list, we have stored 3 different items. So here loop has run 3 times. But if I stored only one item, so here I have stored only one item, so the loop will run only one time because there are no more items. So if I run it, I am only going to see apple, which means the loop has only run one time. You can use any, Zililam, you can use any name you want. Suppose I have used F here, you can also use G. You can also write, suppose if I write fruits. Now see, there is a difference. There it is fruits and it is fruit. So this is my temporary variable, and this is my actual variable inside which my value has stored. So here you can give any temporary variable you like. Okay. Yes, Divya, we are going to do that as well. I'm going to give you one question. In that question, we are going to learn that as well. Ashwini, we are using the square brackets because we are creating a list. Okay, here we have created a list. Square bracket means we have created a list. Suppose if I don't use a list here, then how are we going to store multiple things? Just like I discussed in the starting of this lesson, that you have 100 different DNA sequences. Now to create multiple variables to store those 100 different DNA sequences, now this is a very hectic work. You cannot create 100 variables. So what is the best solution? You can create a list. And how to create a list? You just simply use square brackets. Okay, now inside the square brackets, you can add any items you want. Divya, we are not covering tuple, okay? Today we are not covering tuple. We are only learning about loop and list. Okay, then let's proceed with our practice question today. Here is your question number 1. Okay, you do not need to run the code. Only tell me by looking at this question, without running the code, predict the output. Read this question and write me in the chat box what this line is going to print me, what these two lines are going to print me. See, this is the, okay, wait. Let me see your answers. Okay, 3 and 6, 11 and 6. Okay. Okay, look at here. I've already done the question answer as well. See here, this is a variable sequence, and inside this variable, the question has stored a list. So this is our item number 1, this is our item number 2, and this is our item number 3. So here the question is print the length of sequences, means what is sequences? This is our variable. So what this line is going to print us? It is going to print us the number of items stored in this list. So there are 1, 2, 3. So the answer of this line is 3. Now print the length of the sequences at position number 2. Here in our variable sequences at index 2, 0, 1, 2, this is the length. We are talking about this DNA sequence length. So 1, 2, 3, 4, 5, 6. So the answer is 6. Alright, here we have written print, we have used the length function, then we have given the name of our variable sequences inside which this list had stored, and here we have given the index number 2. Now we know in Python indexing starts from 0, so 0, 1, 2. So we are talking about this DNA sequence, the length of this DNA sequence. So the answer is 1, 2, 3, 4, 5, 6. So the answer is 6. Is it clear? Who has written 1, 3? Okay, Vigneshwari, is it clear? The answer is not 1. Everyone who has answered 3 and 6, very well done. Let's move on to question number 2. Okay, everybody open your VS Code. This is your question number 2. And do not forget to mention your name. You have to create a list called DNA sequences. You have to make a variable named DNA sequences, and inside that variable you have to store a list. Then you have to print the list and you have to print the type of your variable and share your screenshot in the group. Everyone share your answers in the group. Are you following the question? Is there any difficulty? Should I explain? Okay, we have received our first answer. Sudeshna, very good. Print the list as well. You have printed the type of your variable, print the list as well. Okay. Paripurna, very good. Good, Swati. Okay, Aditya, very good. Jumana, share the complete picture. Ananya, write your name in the double quotes. See here, I have written my name in double quotes. So you have to write your name in the quotation marks. Okay, Ananya? Very good, Ashwini. Tisha, you have created a variable called DNA sequence, right? And inside your variable, you have created your list. So why are you writing print D? Write the name of your variable, that's why it's showing error. Okay, first of all, print your variable. If you are trying to use the loop, first of all, read the question. Everybody only do what is asked from you in the question. Okay, Tisha, your error is showing because you have not used the indentation. Also, do not move ahead. We have question for that as well. Just do what is asked from the question right now. We have for that question, loop question, we have that question as well. Very good, Anmol. Okay, let me show you the answer. Everyone, do not move ahead. We all have to move together, all right? Not everyone knows anything. I know you are intelligent, but stay with the class. So, this is the answer. You can refer your answer with this here. So, here the question is you have to print your complete list. So, just make your variable, create a list inside your variable, and print your variable name. So, your variable will print your list, and then print the type of your variable. Okay, let's move on to question number 3. This is question number 3. Do not forget to mention your name. Everyone, we will do the loop question as well. Please stay with the class. I know you are very intelligent, but please stay with the class. We will do the loop question as well. Everyone, do question number 3. Write a Python program to print the total number of DNA sequences. and print the length of the first DNA sequence. Vivek, do not write your fifth line print type of DNA sequence inside that. You have written that inside loop, right? You have created a loop, for loop. So you have written print type of your DNA sequence inside the loop. So do not, so remove your fifth line. So Darshana, very good. Athira, very good. Ananya, very good. Anmol, very good. Anurag, very good. What other DNA sequence are you talking about? Also, please mention your name. Everyone, don't forget to mention your name here so I can't see whose program belongs to whom. Okay, your code is correct. Divya, your code is correct. Okay, let me show you the answer. So the question is that you have to write a Python program to print the total number of DNA sequences. And we can see that inside this list, the total number of DNA sequences are 3. 1, 2, 3. So that means to print the total number of these DNA sequences, we have to use the length function. So here we can write print, we can use the length function, then write the name of our variable. So this line will print the length of our list, and this line will print the length of the DNA sequence stored at index number 0. Sameen, why have you created a for loop? Is the question asking you to create a for loop? Why are you using for keyword? Just only write like this. Here I have written the answer, Sameen. Sameen, remove your third line. Khushi, very good. Okay, so here we understood the difference between the length of this total list and the length of the DNA sequence or the length of the item stored inside a list. Okay, let's move on to question number 4. Here is a question number 4. Some students keep on forgetting to mention their name. Okay, write your name as well. So this is question number 4. You have been given a list. So you have to print the first nucleotide of the second DNA sequence. Now this question might seem tough to you. I think Divya asked about this question. Divya, now you can do it. Let me know how many students can do this question. You have to print the first nucleotide of the second DNA sequence. So this is your second DNA sequence, and in the second DNA sequence, G is your first nucleotide. So you have to print G. How will you do it? You only have to print G. And then the last nucleotide of the first DNA sequence. Okay, second. So this is your second DNA sequence and the last nucleotide of the first DNA sequence. Let me see your answer of this question. Okay, very good, Ronak. Your screenshot is blurred. Krishna, I think, yes, Krishna, your screenshot is blurred. Okay, Vedasri, I have looked at your answer. Okay, Vidasri, take a look at the question again. You have to print the first nucleotide of your second DNA sequence. 1, 2. This is your second DNA sequence and this is your first nucleotide. Means you have to print only G. And the last nucleotide of the first DNA sequence. So this is your first DNA sequence and this is the last nucleotide. That means your first program should print G and your second line of code should also print G. Vidasri, are you understanding the question? Anurag, very good. Aditya, very good. Dwaipayan, very good. Paripurna, very good. Okay, so I'm seeing very less of you are sending your answers. Ashwini, very good. Okay, let me show you the answer. Divya, I didn't see your answer. Okay, so this is the answer. Divya, your screenshot is blur. Share it again. Sudeshna, very good. Yes, it is correct now, Vidasri. See here, Okay, first of all, I want to ask you who can explain this answer. Okay, raise your hand. How many of you students did not get the answer? How many of you didn't understand it? Okay, let me know how many of you are there. Okay, let me explain. We already learned the indexing, that we use square bracket, and inside the square bracket we give our index number. So first of all, let's remove the second index. Okay, now what does this line mean? This line means we have given our variable name DNA sequence, and the one index means 0 and 1, means we are trying to access this DNA sequence. Is this correct? And the second line means we have given the index number 0, means we are trying to access this DNA sequence. We have already learned this. Now the question asked you to print the first nucleotide of this second DNA sequence. To print the first nucleotide means to print G of this second DNA sequence. We have already given the index number 1. 0, 1 means this is correct. Now here we will add one more square bracket, because by writing the index number 1 we have reached here, but to reach at G we have written here 0, because we already know that indexing starts from 0, and inside this string G is at index number 0. So by here 1 means this DNA sequence, and by here 0 means this nucleotide or this character of the string G. Okay. Similarly, in the second line, we have written 0, the index number means this DNA sequence, and to access the G of this DNA sequence, we will write 1, 2, 3, 0, 1, 2, 3, 4. So here we will write 4. Okay, so here 0 index means this DNA sequence, and here 4 means the fourth nucleotide, 0, 1, 2, 3, 4. So the last nucleotide of this string. Or we can also write -1 as well. We learned yesterday that there is negative indexing. As the first character is always on 0 index, the last character will always be on -1. So we can also write 4 in the positive indexing, or we can also write -1, as the last nucleotide or the last character will always be on -1. Is it correct? Is it okay? Yes, Anurag, negative indexing we learned yesterday. Okay, Samin, I will explain it again. See, here we have created a variable DNA sequence, and inside this variable we have created a list. Today we learned that in a list these are called items. These are multiple items, or these are multiple values of a list. So here this first DNA sequence is on index number 0. The second DNA sequence is on index number 1, and the third DNA sequence is on index number 2. So the question says that you have to print the first nucleotide of the second DNA sequence. So this is our second DNA sequence, correct? This is our second DNA sequence, and we know that it is written inside quotation marks, means this is a string, and yesterday we learned indexing in string. Let's copy it. So here, let's copy it. And here I'm writing here. Let's give it some spaces inside this. Yesterday, we learned indexing in string, that G is on 0 index, then this G is on 1, and then 2, and then 3, and then 4. This is the indexing we learned yesterday, that in a string, each character has a specific index number. So here we are seeing that we have to print the first nucleotide. So here in this string, the first nucleotide is G. So it is on index number 0. And in list, this DNA sequence, it is an item which is stored on index number 1. So by writing 1, by giving the index number 1, we are saying the Python go to the index number 1. So this is index number 1. This is the DNA sequence stored on index number 1. So 1 means this DNA sequence. Now we have to print G. So here we know that G is on index number 0. So we have written 0. Okay, so 1 means this DNA sequence and 0 means this G. Okay, is it clear? Is there any confusion now? Tisha, what will be 5? Divya, is there still any confusion? Okay, 0, 1, 2, 3, 4 and 0, 1, 2. So why will it be 5? Prachi, I will come on that. What I just said now, is it clear? Yes, I will come on -1 part as well. Yes. So similarly, the second line said to print the last nucleotide of the first DNA sequence. So this is the first DNA sequence. It is on index number 0. So here I have written 0. Now we have to print the last nucleotide. So here the last nucleotide is G. Now let's copy it. Let's give some spaces here. Now we know that A is on index number 0, T is on index number 1, then 2, then 3 and then 4. So the question says that we have to print the last nucleotide. So here G is on index number 4. So here 0 means this DNA sequence, this string, and the 4 means G. Okay, so the 4 means here G. Now, this is called positive indexing that we learned yesterday. Now, what is called negative indexing? Here we are going from left to right. In the positive indexing, we are going from left to right. In the negative indexing, we go from right to left. We talked about number line yesterday, that in maths there is a number line. In the middle there is 0 and after 0 there are positive numbers 1, 2, 3, 4. Before 0 there are negative numbers -1, -2, -3. So just like we learned that the first character will always be on 0. No matter which character is there can be T. Suppose there can be T or there can be G, whatever. Anything can be present here. But the first character will always be on index number 0. Similarly, the last character will always be on -1. So here, in the negative indexing, G will be on -1. Let's make it a comment. Okay, so here G will always be on -1 because it is the last character. So it's -1, it's -2, -3, -4, -5. So here even if I write 4, it is representing G. Even if I write -1, it is still representing G. Okay, is it clear? Okay, Mohammed, is it clear? Prachi, is it clear? Just like the first character will always be on 0 index, the last character will always be on -1. Vidasri, just like I said, in the number line, in the middle there is always 0, right? And before 0, there is -1, -2, -3. So there we are not using the 0 again. 0 will always be used only one time. So here in the positive indexing, A character will always be, or the first character will always be on 0 index, and the last character in the negative indexing will always start from -1. Then it will continue -1, -2, -3, -4, -5. Okay? Yes, Sahan, you can use anything. You can use positive indexing, you can use negative indexing. Okay, let's move on to our question number 5. This is the last question. Now you all can do loop. Write a Python program to print each DNA sequence using a for loop. Your favorite question, every one of you were doing loops. Very good, Sudeshna. Very good, Ashwini. Paripurna, very good. Aditya, very good. Anurag, very good. Snekha, very good. Sameen, very well done. Divya, in your third line, you have written print DNA sequences. You have to write print I. See, in your second line, you have created a temporary variable named I. Because every time the loop will run, the first item will be saved in your temporary variable. So you will have to print your temporary variable, okay? Divya, is it clear? Athira, very good. Azmi, very good. Also understand the concept how loop is run. Yes, it's correct now, Divya. Very good, Neha. Let me show you the answers. So you have to write a program to print each DNA sequence. These are DNA sequences, so you have to print it using a for loop. So here we have created a variable DNA sequences, and inside this variable we have created a list, and inside the list we have stored our multiple DNA sequences. Now to create a loop, we are using how to create a for loop. So we have used the keyword for. Then we made a temporary variable name sequence, because whenever the loop will run, here there are three items. That means the loop will run three times. So in the first loop, the sequence value will be ATGC. In the second loop, the sequence value will be CGTA. And in the third loop, this variable sequence value will be TTA. And when we will run it, we will see all the items in this loop in our output. Okay, everyone, very well done. Next week, From the next week, we will finally start working with Biopython as we have made our foundations clear in Python. Okay, we will start working with Biopython. So I will upload the assignments related to all the lessons we have completed till now. So keep doing your revision and also complete the assignment as well. Okay, I will upload it in the Google Classroom.